and we talked about, so far, we talked about the immune mechanism, and we talked about hypersensitivity when where your immune system overreacts, right? Now this is another clinical situation. If you go back to the purpose of immune system is for self-defense, right? Now in the process of self-defense, the cells are equipped with killing mechanisms, whether they be cells, proteins, and antibody. It so occurs that sometimes the friendly fire, or more than friendly fire, they kill their own cells. There is a possibility, right? And that leads to a mechanism which we call autoimmunity. Your own cells start killing you. Right? So this is what you have to understand. And there are quite a few diseases. You know, what can you do if your own cells start killing you? We have to understand the mechanism. Two mechanisms. Remember, if the cells, how do they identify? Because one of the purpose was to identify self from non-self. If you fail at that level, if your cells are unable to identify, right, you just took a person, your friend, as an enemy, so you kill it. That's what the cells do. There's a possibility. We have to understand so we can come up with better drugs. That makes sense? So today's lecture, if you understand why do we have autoimmune diseases. And one of the most famous autoimmune disease, they think, is diabetes type 1. So they think that your own cells are killing your own pancreatic cell, leading to a disease, so on and so forth. Right? Now, I'm gonna give you an idea. Now, one of the things, if you remember classically, that when these T cells are made, right? When we are in the womb of the mother, T cells and B cells are made. So there is a tiny organ that disappears at birth. It's called thymus. It's in the pregnancy, you know. So these T cells, when they are made, so these T cells are given a, what should I say, analogy-wise, a barcode reader. They are given the barcode reader. The machine is going to read all the cells. And remember, I told you each and every cell has a barcode. So they are trained that, look, you are the cells, right? And this is a SS number, or we call CD number, of each and every cell of you. So put it in your memory that if you come across these cells, don't react. We call this education. We teach them. It's called thymic education, right? But it's a biological system. System can fail. But if the system fails, then you start killing yourself, right? So what it does is that it gives that information to the T cells in the thymus, right? And make sure they are trained for that. If by chance they are not trained for that, so the thymus actually will make sure that those so-called self-killing cells are eliminated. The thymus itself kills them. If they're unable, it's just like you pass and fail. If you fail, you're not promoted. You're done. So they test, thymus tests them and say, you know, what do you think of this cell? Is this you or somebody else? If thymus, the cells fail the test, kills them. Because you don't want those cells who can identify between cell versus non-cell. They're not supposed to be there. You understand? So this concept is here. So what it does is, for example, when these cells are made, and there are cells in thymus which are called thymocytes. Thymocytes are special cells in thymus, right? And then these cells usually are either CD4 or CD8, we already know that, okay? And then the cells in the thymus, we have this antigen presenting cell, right? And this presents a particular antigen in contact with class 2 or class 1, your gene, is in the gene, okay? And present them to these cells. Okay? Now what happens is that it so occurs that some of the cells, for example, which are T cells, and they have very high affinity attachment with their own cells in terms of their own peptide, right? Right, they kind of hug them and bring them closer. So there is a chance that they may be a problem because they should identify and then separate. If they kind of have this covalent bonding for a long time, right? So they have a very high affinity for this one in terms of attachment to their own self. So these will kill. It's called negative selection. They are killed. Okay, and those cells which have a low or intermediate affinity, right? They are left to survive, whether they are CD4 or CD8. So this is one of the mechanisms that you just want to see how tight the bonding is. If it's very tight, there's a danger that they're going to overreact. Okay? Can you bring my book as well, Malik? Just want to make sure that I'm not missing anything because I didn't see the legend. Is there any question on this concept? Does it make sense to you? Important thing in this one, which is another concept, which we call it's called mechanism of central tolerance. Mechanism of central tolerance. So in this case, you have to keep in mind that there are two types of antigens that your system is going to see. One of the antigens are cell-bound, they are attached to a cell. We call cell-bound antigen. So all the cells of your body will have a antigen or will have a barcode for them to be identified. Or there could be a soluble self-antigen. So self-antigen means that yourself, all the cells of your body, remember I told you each cell of your body is unique to you. And your cells are unique to identify that. And they have been trained and educated not to kill your own self, right? But if these self-antigens are there, whether they're attached to your cell or they're moving out freely, what happens? So what normally in this case, what happens is that if there are, for example, in this case, there are B cells, for example, this one over here is a B cell, this is a B cell, right? And if you remember, when we talked about the antigens, B cell is an antigen presenting cell, okay? And this is going to react with either of these, right? And when they come with the cells which are B cells, but they can identify, because these were the T cells. Remember, over here we were talking about the T cells. Because these T cells were in the thymus. That's what T for thymus, T for T cells. In this case, if you find some B cells which are self-reactive, right? They react with your own cells. So what does the body do then? Because we need B cells, but we don't need B cells, we start killing you. So what we do is, we are going to delete these B cells, get rid of them because we don't want them. Okay, one. Or we just want to make them inactive. Inactivity of the cells is called anergy. Anergy. An means absent. Right? So anergy means not energy, anergy means no response. Or sometimes what body does it says that, okay, these are the B cells which have a receptor which react with your own cells, right? It's just like, you know, 5 of you and I ask you your cell number and you give me the same cell number. That's not possible, right? Just imagine if T-Mobile is going to give same cell number to 2 people, so there's a problem. Normally it doesn't occur, but if there's a problem, so you say, okay, wait a minute, we're gonna give you a new number. So this will get, we call it a receptor editing. So if your receptor acts with your own cells, we wanna change this receptor. We call it receptor editing. So that your number is unique. It's not messed up with anybody, right? And then we want to make sure that now these B cells should not react with T cell. So the idea is that your B and T cell, which are lymphocytes, should not be killing your own cells. But if they do, there's an issue. So this is how the issue comes. Why we do have disease. The disease is that this is the ideal mechanism, but some of this T cell escape this scrutiny. And some of the B cell will escape. So these are the B and T cell escape the mechanism of testing, and they are sitting somewhere. And after some time, they come back. They're hiding, and they come back, and they start killing you. And then you have autoimmune disease. So far, so good? Does that make sense? Now, let's move on. Now, in this case, what's happening is that that concept over here that you don't want the B cells to respond, right? So B cells don't respond to your own self. We call this concept energy. But now the question is, how does body do that? How does the body have energy? Because we have to learn that. So what body does is that if you remember a classical definition for B and T cell, typical immune response. It's a typical immune response. Immune response. So the T cell presents an antigen to a B cell. Okay? In this case, there are a lot of receptors, but these 2 receptors, if you remember I said these 2 receptors are important because these 2 receptors are important for cytokine synthesis. Cytokine synthesis. Right? So they see, and then again, there are 2 signals, 1 signal and 2 signals. So these signals are required before this B cell gets activated to make antibodies. So this is like, I give you an example that normally, you know, like if I want to go to my office, I'll just have 1 key, I'll open it up. But if there's something very secure, or you do look at bank locker, or so they will add 2. We just want to make sure security. So we have 2 signals over here because we don't want B cell to activate and start making antibodies. This is normal procedure. Right? That makes sense? It's a typical thing that we already do. So signal 1 and signal 2. So you can, signal 1 is provided by the antigen binding to a co-stimulative linking the BCR. So this is signal 1 over here. Signal 1. And signal 2 is provided by the interaction between the CD4. So this is signal 1 and this is signal 2. So this is signal 1 over here and signal 2. So both are required to get activation to produce antibody. This is normal. And why we are talking about that, if you remember the other day when we talked about you develop antibodies to your own cells in neuromuscular junctions against the receptors which are for acetylcholine. And acetylcholine comes and the receptors are already bound by the antibodies. So there is no acetyl binding, and if there's no acetylcholine binding, there will not be any contraction. We talked about that the other day, right? Leading to hypersensitivity reaction. So this is, now what happens is that if one of these signal is missing, right, in the absence of this second signal, there is no co-stimulatory signal. So this means there will be no activation and no antibody secretion. And why I'm saying it? Because if your body starts making an antibody, in that case to your own acetylcholine receptor, you want to stop that. How would you stop that? So you want to make sure that you inactivate this co-stimulatory molecule, right? Because then there will be no reaction. So this is one of the treatments that we have, right? That's number one. The other thing is that the B cell will always need help from T cell. It needs help from T cell to make antibodies. Okay? So if you don't have this signal from T cell, there is no help, and B cell will be energized, not energized. This means no reaction. So that's another thing. So you can have two types of drugs, actually three types of drugs. Either block this signal, number one, block this signal and there are drugs available, right? Or block this antibody. So these are three possible treatments of autoimmune diseases. So far so good? Does that make sense? Okay? Now in this case, that was one case for the B cell. This is a case of co-stimulation of T cell. So this was T cell energy. This was B cell energy. This was B cell energy. So how you make B cell becoming energized. And this one you have T cell energy. Because you can either stop T cell or stop B cell. So in this case what's happening is same story. So you have an antigen presenting cell which presents this antigen over here, right, to a T cell with two signal. Over there the signal was CD40, and this is a signal, right, which is a co-stimulatory molecule. Co-stimulatory molecules, right? And it is going to make the activation of T cells, and then the activation of T cell is going to secrete cytokines, right? And the cytokines are required to regulate the whole immune response. Okay, so what can you do? So what you can do in this case is that, again, you can either, this is overhead signal 1 and this is signal 2. Right? So what you can do is that if there is no signal 2, like in that case, then the T cell will have no, this B7 and B8, this is not there. So there is no B7 and B CD28, so no reaction will take place. And then you will have T cell energy. So the idea is that of the two signals required, it's just like I gave an example, you have two keys, but you have one key is not going to open the door. As simple as that. Whether it's a B cell or it's a T cell, or as a matter of fact, any tissue cell. Right? So you can see from here, two signals are required for T cell activation. Signal 1 is provided by the recognition of peptide plus MHC, peptide plus MHC. Signal 2 is provided by the interaction of the co-stimulatory molecule B7 expressed on the surface of an APC such as dendritic cell and with CD28, which is on the T cell. And if both of the signals are there, the cell gets activated and does. But in this case, we can stop that if the cell is going to activate against your own cell. How do we go back in drugs? So in this case, again, you can see we had, and you can Google, I'm not going to ask you, this is like first line of drug. You can block CD40, you can block CDL, second line of drug. You can make an antibody for the third line of drug. for B cell. In this case, what you can do is that you can block this receptor over here, first line of drug. You can block this receptor over here, and this actually is available. CD20 blockers are available as a drug. One of the autoimmune diseases is rheumatoid arthritis. In rheumatoid arthritis, the treatment is given to block CD28. It helps. It's a drug available. Very common drug. If you go and Google it, and I'm not going to teach you because that's a little bit too much beyond the course, but this is what we have. So you block that so that cell get not activation. But you can also see the problem is that if you block everything, there will be no immune reaction. The very first thing that you will have will infections. It's just like say since there is a friendly fire by the police, so no more police required. What's going to happen? Right? So there is a danger, but you have to control it. But there are pitfalls in everything. You understand the concept for signal 1, signal 2 concept of autoimmune diseases, how we block them. Right? Now the last concept over here is that what are the other possible mechanisms of triggering autoimmunity? So the cells are there, whatever they are there. But the question is, most of the diseases of autoimmune diseases, they show up late. Firstly, they are in women, and they comes in 30s. So they start wondering, something to do with the gene, something to do with gender, something to do with late age. But what triggers them? Then they trigger. So why would not a woman show a autoimmune disease at the beginning? Why she shows so there should be some triggers. So what they think, there are many mechanisms, but they give at least 2 mechanisms or 3. What was the third one? Did you write?
What's the third one? That one was a part of A.
Sure. So what happens is that now, so we already found out that there are B cells that are already hidden, right? And they're autoreactive, but they do anything. So what triggers them? So what we found out that these cells basically did not activate because there was no T cell help. We already discussed that, right? So in this case, the typical example is that some of the microbes, for example, some of these microbes, right, they come up with, they kind of hijack the antigenic nature of your self, right? So for example, in this case, let's see, if your self-antigens looks like this, and it mimics, it's a mimicry, so your microbial antigen looks like this, right? So this is an overlap. So if this is designed, it's just like I'm just going to put my number, if it says a amino acid sequence of, this is my office number, so everybody knows it. So if this is your self-antigen, right? And the microbe also carries the same antigen. So it's going to trigger that, right? You know why? Because what happened is that your body will think that this is a microbe, right? And these microbes have to be dealt with by making an antibody against the antigens of microbe, but it shares the same nature of your own cell. Like for example, I'll give you an example for that as well. For example, let's say strep infection. They said, did you have any strep throat infection when you were a kid? The very common question asked. Because your body makes an antibody against that strep. That antibody also recognizes the cells of your heart. So they kill that bacteria, but in the process, they also kill your heart cells. Rheumatoid heart disease. right? That's why strep throat infection in the kid is taken very seriously. They go for the test. Group A streptococci. It has to be treated, because if you don't treat it, you will develop an antibody which is similar. So the strep B bacteria has the same code, kind of, that will lead to an antibody which will be destructive against your own self as well. So this is one of the ways that autoimmune disease work, right? Because in a typical situation, you can see from here on the picture as well, that the T cell will have to activate B cell by this mechanism, right? And then the antibodies will be made. Okay, so in this case, the antibodies are made which are microbial. You can see from here, the color microbial antigen antibody over here, this one, right? And then the antigens are made for your self antigen as well, which is over here. Okay, so this antibody is going to kill the microbe for which it's made, but it's all going to kill you as well, because they share the same molecular structure, which is called molecular mimicry. You got an idea? So one of the things is group A streptococci, we call gas. Group A streptococci, especially in the kids. If you go and see a patient of rheumatoid heart disease, right? One of the first questions you ask that, did you ever have a group A streptococci infection when you were a baby, and was it treated or not? So if not treated, so you made an antibody that killed the bacteria, but in the long term, it's going to ruin your heart as well, especially the valves of the heart, leading to rheumatoid heart disease. That's the mechanism. That's why you shouldn't take sore throat lightly in kids. And you can't say it's viral. They always test for group A streptococci, because if it's a group A streptococci, you have to rigorously treat with an antibody. Otherwise, you'll make antibodies, like in this case, where this antibody will recognize your self-antigen present in the heart or elsewhere. That's what they say, the antibodies go and kill the beta cells of your pancreas, which produce insulin, and you have a type 1 diabetes mellitus. So that happens. That's one concept. The other concept is that lipopolysaccharide, right, which is also an endotoxin. If you remember your microbiology, so this is present in almost all bacteria. Yeah. So this, when body produces tons of antibody against lipopolysaccharide. So what happened is that in producing that, it can produce many clones. Clone A, clone B, clone C, and clone D. And it may happen that in one of the clones, the antibodies are self-reactive. If you have self-reactive antibodies, this means they are going to react to your own cells, whether they be in heart or kidneys. So many a times your heart cells or kidney cells or any other organ cells are damaged by the antibodies produced by your own self in the process of fighting an infection. So one of the things that you can see that the possible mechanism may not be true is that you trigger autoimmunity, right, by getting infections. So one of the processes is that infection leads to autoimmunity. So the idea is that if you get infection, you try to make antibodies, and in making antibodies, you make antibodies, especially the clone, which, you have a clone A, clone B, different type of antibody, clone D, but one of them also becomes clone C, which is going to cause damage. So you damage your own cells. If you damage your kidneys, you get kidney disease. If you damage your pancreas, you get pancreatic disease. If you damage your neurons, you get, because these antibodies are going to go and kill the tissue cells. So that's the concept of autoimmunity, and that is the concept of friendly fire. And then the question will come that how does body protect? Tolerance, that's another mechanism. Tolerance, how does body tolerate? Because there are too many antigens are overloaded to us. So we'll discuss that tomorrow. What are the other mechanisms of tolerance? Okay, any questions?